Welcome to the 2022 Target ALS annual meeting.
Thank you very much.
Are you enjoying it so far?
So far, absolutely. It's been great. Obviously, having the opportunity to meet my colleagues and friends after 2 years is well appreciated.
What do you think over the last few years about we've just spoken about the very recent developments, but we've been around for about 8 years. What do you think of the developments over that time and what Target ALS has brought to our community?
So that's a fantastic question, and I think that, it really has come together. Right after the initial investment and Target ALS's mission, which they were committed to, right, you now are seeing the synergy and what that mission is all about, and that's bringing together academia and industry, and the buy-in is now there. And so I actually saw that for the first time. I mean, arguably when Target ALS was first established, I would probably say there was a little bit of resistance, like, is this concept really going to work? But now, after the seeds have been germinated, right, I think that people are really now finally are understanding, like, I really can see what Target ALS is about. They're trying to do something that's meaningful for the patients, and that perhaps this is the only way that it's going to be accomplished. And obviously the annual meeting that I think brings together those individuals from both industry, academia, foundations, funders, VC groups, and the buy-in is there.
What are your thoughts about how the scientific cores, and for those watching at home, what a scientific cores are resources and tools that we create specifically based on researchers' needs, and it can be a wide range of resources. What do you think about, it's sort of half of our model, right? We do collaborative funding is the big part, but we also, the other half are these scientific cores. What do you think about that?
I think it comes down to enabling, right? And what these cores do, whether it's for a particular resource, for biofluids or postmortem tissue, right? Or even today, one of the talks, the latest initiative was, greater accessibility to animal models. And one of the animal models that we developed, are now, being distributed through Target ALS in partnership with Jackson Labs, and to have an investigator be able to test their drug target using my mouse model and being conducted, through a very, rigorous process with Jackson Labs is gratifying, right? I feel like I'm now part of that, mission where I've enabled an investigator at University of Michigan to do what he wanted to do, right? And that's cool. I mean, there's no other way to say it, but it was awesome. Say, well, you know what? I wasn't part of that, but actually I am, in a behind the scenes type of way. So I think that's the core mission, right? It works.
We at Target ALS, we've got a partnership with AFTD, exploring treatments and biomarkers for both ALS and FTD. We have a partnership overlap for ALS and Alzheimer's. That could confuse people. They will think they're very different diseases, right? So what is it in the research, and why do we do that? Why are we researching multiple diseases and the overlaps?
Right, so unfortunately what happens is that some of the similarities, which I think is probably a strength because you're bringing in investigators from multiple areas of expertise, but it's the genetic overlap and the pathological overlap, right? The same pathology that you see in an ALS patient also presents in a patient with FTD. Some of the genes that we study are present in both. And so, it represents a commonality, but at the same time, perhaps a very complex question is like, how is it that someone who has the same mutation, take chromosome 9, how is it that they have an FTD phenotype, but another individual who has the same mutation get ALS? And we really need to understand, more of that. But I think it's, once again, the common genetic overlap and pathological overlap, between diseases, including Alzheimer's disease as well.
Sure. I want to ask you about a buzzword as my final question.
Go for it.
So, biomarkers.
Yep.
So, It's a word that I've heard it today, I think, 100 times,
Yep.
right? What can you tell me about biomarkers and once discovered, what will it do for us?
So a biomarker, comes in multiple flavors, right? So you have a biomarker that might be able to diagnose someone. Does this person have ALS? You might have a biomarker that can be detected in either in cerebral spinal fluid or plasma, and it may be able to determine the prognosis of that individual, right? How severe will the disease be? How long and what have you. You also will have a biomarker that is important for target engagement, right? So industry, biotech company makes a drug, and they need to know whether or not their drug is really working in the brain. And so therefore you would need something that can be detected with great reliability to see if there's that target engagement that's involved.
Sure.
So biomarkers aren't going to go away because they're immensely helpful not only for the clinicians, and we need better biomarkers, but also for industry because whenever they develop a target, they're going to need some sort of marker to make sure that their drug is working.
Yeah, sure.
So, you mentioned FTD, and I think that raises the complexity of an example of why a biomarker is needed. So, an FTD patient can present with a TDP-43 a presentation or pathology, or with a tau, phenotype, which we determine FTD tau and FTD TDP. But to the clinician,
sorry to interrupt.
Sure.
Explain. So those proteins are.
So those proteins abnormally accumulate in the brains of these patients, and when they come into the clinician's office, they're clearly two distinct pathologies. But yet the clinician is unable to distinguish them from a clinical perspective because they present fundamentally the same way. So imagine if we have a biomarker that will allow us to determine if that person has an FTD tau or an FTD TDP-43, because then you could put them in the appropriate clinical trials when those therapies are developed. Because you could not group that entire population into the same trial. It wouldn't work.
Yeah. Thanks, Len, for your time. I really appreciate it.
My pleasure.